morning in a relatively short period of time. Although overlap, there's going to be it's picking up a little bit where Dr. Glarder left off and overlapping a bit. Yeah. Let's see. Is that better? Okay. All right. Thank you. The same disclosures as the last three lectures. So here's where we're going to pick up again. And I'm going to review a little bit some of the things we talked about in the last lecture. We talked about these complicated concepts of recombination and linkage disequilibrium, and you're probably wondering a little bit of why did I bother torturing you with that. And hopefully I can convince you there's some use to that when we talk about doing genome-wide association studies. I know Dr. Glarder's touched on this somewhat, and I'm going to expand on it a bit. And then I want to end up by talking about personalized medicine and what we mean by that, and sort of where we're going to be going in the future and what you're likely to see, the impact of all this great genetics in the lab, where it's really going to impact in the clinic, and where you're likely to be using it in the future. So let's start with that. What can we get out of all this genetics? You know, at least I'm convinced this is fascinating stuff, and I'm spending a lot of my life working on this in the lab and figuring out what genes cause what diseases, and all this kind of stuff is great fun and really exciting and interesting from the scientific point of view. But what does it really mean for patients? So if we find the gene that causes a disease, what difference is that going to make for the patient? Well, one thing, sort of the first thing that happens once we have a gene in hand, particularly nowadays with modern technology, is it gives us the ability to make an accurate diagnosis. And just making a diagnosis is of enormous value in lots of clinical settings. For the Mendelian diseases, the diseases due pretty much just to one single gene. Hemophilia, familial breast cancer, if you get the gene, you've got the disease. If you don't, you don't. Once we know the gene and we can find the mutation to test a patient's DNA and figure out whether or not they inherited it, whether or not they're going to have this disease, our diagnosis, it's far better than almost any other test you're going to use in clinical practice. If you have a family at risk for Huntington's disease and we test a patient for Huntington's disease, we will tell pretty much with 100% accuracy that they have the disease or they don't have the disease. We'll be able to predict pretty much with 100% accuracy that they're going to have this condition or not. So that's extremely useful and an extremely powerful test. For complex diseases, as you were just hearing about, heart disease, diabetes, things that have a genetic component, but it's not everything, and it's not a single gene, it's many genes. What we can tell from diagnosis here is a little bit less straightforward, and we're going to talk a fair bit about that. Well, can we use a specific genetic diagnosis to guide our treatment? And here we're still in our infancy. There are some examples. To a very limited extent, you know, we can subclassify some cancers based on the set of mutations they have, but this is sort of just beginning. And there's this hope that we're going to be able to customize our treatments, make designer treatments, choose the drugs that patients get based on their genetic makeup. And we'll talk a bit about that, but all of this, I think, is more promise than reality at this point. So here, for sure, I would say that what we've accomplished in genetics and in medicine has vastly exceeded most of our expectations. Again, as I'll tell you, we've got probably about 5,000 genetic diseases for which we know the responsible gene. We can, at least in theory, precisely say that this patient has this disease or doesn't have this disease based on the genetic test. And when I was a medical student, this was beyond even fantasy at that point, that you could do this. So I think we've vastly exceeded expectations. But I have to say that for most of the rest of these impacts of genetics, I think we're far below expectations and what we've promised. And this sort of always troubles me a little bit. If you just read the newspapers, you know, if you just read the New York Times and USA Today, you would conclude that cancer not only has all cancer been cured, but it's probably been cured 4 or 5 times over. We don't have to worry about any of the diseases anymore. In reality, mortality for most cancers has not changed that much over the past 20 or 30 years. So I'm humbled by that as a physician scientist, that we tend to get very enthusiastic about a new discovery, but it's not so easy to reduce a discovery to real practice where we can really clinically impact patients. We can diagnose now better and better and better, but it's doing something about it that's still pretty tough. Okay, well, we talked about positional cloning. This is why one had to know about linkage. When we could take markers, of which we now have, these are common variations among everyone in the population. We can use these as road signs to trace the inheritance of parts of a chromosome and figure out where a gene is. We now have 23, 25 million of these through the genome that we can use. And by using just a limited set covering all the chromosomes, we can easily figure out in a family which part of which chromosome is being inherited, along with the disease telling us that the gene has to lie there. And it's now a relatively straightforward business to sift through the genes in that region, figure out which one has changes in families with this disease, and know what disease you're dealing with. So in practice, if you ever encounter, and there won't be many of these left by the time you get into practice, but if you go to a clinic and you see a patient who has a very strong family history and you think this is a single gene disorder in this family that's not previously been defined, all you got to do is collect a couple of affected individuals, and it's now possible to get here fairly quickly. And this is not all just theoretical. Actually, last time I was in clinic, the time before last, I saw a family, a patient was referred to me with a very low platelet count, otherwise perfectly healthy. Has had this most of his life, it seemed. And not only that, but he had 4 siblings, all of whom had low platelet counts in the 30 to 70,000 range. Low, but not all that low. His daughter had the same thing, and his granddaughter had the same thing. And no apparent cause that anybody could figure out. And we're now in the process of doing what one does now. We don't bother with most of this. We just go straight to sequencing the whole DNA and seeing if we can figure out where the defect is. And there are still some families out there. It's usually fairly rare diseases. If it's not been, if there are a bunch of families out there, probably someone else has already done it. This is a compilation you can find at NCBI. Now, this is from a year and a half ago. If you go and look now, this number is closer to about 4,000. This is continually rising, the rate at which we're identifying the molecular cause for a Mendelian disease. And I think within a relatively short period of time, it's already largely true. If a patient has a single gene disorder, a genetic disorder due to a single mutation in a single gene, it's probably already known, probably already figured out, and it is possible to make a precise molecular diagnosis at the single mutation level. Well, so I didn't think I'd ever be saying this, but I think Mendelian diseases, the genetic cause, the primary gene responsible for Mendelian disease, is largely solved at the molecular level. And this leaves us with a very, very large challenge, and this has become the major focus of much of the genetics research community, and that's complex diseases, which you've already heard about, diseases that are not so simple. They may be sometimes due to a single gene disorder, but that's rare. As Dr. Galerder told you, for many of these diseases, a few percent of patients at most will have a single gene causing it. For most patients, it's more complicated than that. There are many diseases. They have subtler changes in many different diseases that together, along with chance and environmental effects, determine whether or not they're going to get this disease. Yet, from twin studies and other kinds of genetic analysis, we know that these diseases have a very large genetic component. They're highly heritable. For most of these diseases, anywhere from 50 to 75% of the overall risk of having the disease is determined by your genetic makeup. If your identical twin has it, you have a very high chance of having it as well. So much of this is determined by your genes. But can we figure it out? It's not a single gene. And linkage approaches basically won't work here, because it's a different set of genes and different in every family, and you're never going to get a large enough family where you're going to be able to sort this out, and this largely failed. So what else could we do? And there are basically 2 approaches that have been taken over the past 15 or so years. A huge effort went into this, candidate gene association studies, which I'll tell you were turned out almost all to be wrong. There are about 35 to 40,000 reports in the medical literature of this kind of study. And we now know from genome-wide association studies that we're going to talk a bit more about that most of these are wrong. At least 90%, and I think more like 95 to 99% of these are wrong. Well, what is a candidate gene association study after I've built it up so much for you? You're all dying to know. What we did here, and it's instructive really, is that scientists, physicians, researchers would pick their favorite gene and look for a common variant in that gene, and then just simply look at a group of patients that have the disease and compare them to a group of patients that don't have the disease. And the hypothesis would be, well, if this SNP, this G to A change in my favorite gene really has some effect on the disease, then the G allele ought to be more common, if that's the bad allele, ought to be more common in people who have the disease than in people who don't have the disease. So if I just collect enough of them and do this analysis, I'll find it. So, for example, let's say you work on the LDL receptor, which is involved in regulating cholesterol levels. So you have a hypothesis. Maybe there's some, I've got this very common, 50% of people have a G in this position in the LDL receptor gene and 50% have an A. So I hypothesize that if you got a G or an A here, it's going to really make a big difference in whether or not you're going to have a high cholesterol. And you now get a whole bunch of people with high cholesterols and a whole bunch of people with low cholesterols, and you look how often do you find a G in one group and how often in the other. And is it statistically significant? There are lots of biases here. One big one is publication bias. And this is a problem you have to think about not just for genetic studies, but for any clinical treatment or does this drug work in this disease or not? If you do a study and it works, you are much more likely to publish it and have it published than if it doesn't work. People get much less excited when you say, I tested out my hypothesis, this variant in the LDL receptor, and it doesn't have any effect at all. Well, journals don't like to publish that, as opposed to, wow, this had a big effect. This thing is important in cholesterol. So positive studies get published, negative don't. And so if you imagine what a p-value of 0.05 means. That means if we did this experiment 20 times, we'd expect to see this difference once by chance. Well, if we do this study 20 times, and one of them is significant at the 0.05 level, and that one gets published, and the other 19 don't, that's publication bias. And that happens a lot. Here's another big problem. Let's say we've got 20 different variants in the LDL receptor that we kind of like, since that's our favorite gene. And we study all 20 of them in our patients, and one of them looks significantly different between the two groups by chance, p-value of 0.05, and the other 19 don't. We're going to publish that one that looks significant, and we're going to ignore the other 19. And this happens all the time. One of the big killers turned out to be population substructure, and I'm not going to spend a lot of time talking about this, but subtle differences between your patient population and your control population. Let's say you're studying diabetes in parts of the Southwest, and it turns out to be pretty frequent among Pima Indians, and more so than in other populations. So among your patients, there's more Pima Indians than among your controls, and you don't recognize this. Well, any genetic variant that's more common in the Pima Indian population is going to look like it's associated with diabetes, and that's a huge explanation and a big problem. So genome-wide association studies is something different. Here we have no hypothesis. Our only hypothesis is that there are some genes, some variants somewhere, and we look at millions of variants throughout the genome all at once and comparing them in our patients and our controls and look to see whether there's a significant difference. And this is now possible because, again, we now have many, many tens of millions of variants that we can look at. But more importantly is this fact, this pesky old linkage disequilibrium that we talked about last time. The fact that our genome is in these blocks that have where the variants are kind of being inherited together, that they like the hemochromatosis in A3. If we did a genome-wide association study with A3 for hemochromatosis, it would be positive. Not because A3 causes hemochromatosis, but because it marks one of these blocks that carries the actual hemochromatosis gene and the actual variant that causes hemochromatosis. So the theory is very complicated, but the bottom line was using about 1 million SNPs, it's possible to make sure we've got ones that pretty much pick up all these blocks throughout the genome. So if there's any block like this that carries a common variant that affects your disease, even though that variant may not be on your chip, a couple other variants in tight linkage disequilibrium with the key variant will be on your chip, and you'll see an association. So what that means is when you get a positive association from these millions of SNPs, the SNP that gave you that association might not actually be the one that causes the susceptibility to disease, but it's near or close by to one that does. So as a result, what's basically done here is you take a large set of patients who have your disease, a large set of controls, and you're very careful to try to match them, do away with population substructure problems, and then you just simply use a chip that's got about 1 million or so common SNPs, and you just genotype all those SNPs in all the patients and all the controls, and then you do a lot of math. And it's very complicated, and most of us don't understand it, and there's a whole new field of computational biology of people who do understand this. And if any of you really like computers and statistics, this is a gold mine. People who can do that stuff well are worth their weight in, I don't know, titanium or something like that now. In the research community, there's a real shortage. But they figure this out and do the math right, and I'm convinced we're doing this right now. This was the first set of things to do, and the first big hit, actually, was in 2005. It's not that long ago. A disease called adult onset or age-related macular degeneration. It's a very common cause of blindness. And lo and behold, what did they find? A common variant, if you have a histidine instead of tyrosine at position 402 of the complement factor H gene, that has a big effect on your risk of getting this age-related macular degeneration. Now, not everyone who gets the histidine gets it, and not everyone who gets the tyrosine is protected. But if you get the histidine, you have a 2 to 4-fold higher risk of getting this disease. And that's pretty significant. It accounts for maybe 20 to 50% overall of the risk of age-related macular degeneration. So remember these. The odds ratio, the relative risk, is 2 to 4-fold. And this accounts for 20 to 50% of the risk. Now, as I'll tell you in a few seconds, almost all the other things we've done are much smaller effects than this. Typically, most of the hits from GWASs have relative risks of 1.1, 1.2, 1.05, and they account for 0.1% or something of the genetic risk. So this is a huge effect. And clinically meaningful. Is it being used in the clinic? Not all that much, because at the end of the day, you're probably not going to do anything until you actually see the macular degeneration. Knowing a little earlier that the patient's going to get it, there's not that much you can do yet, but someday there might be. Well, this was the next big one. This was sort of the holy grail of GWAS studies of complex traits. Type 2 diabetes, as you know, incredibly common. An epidemic, actually, in the Western world. 15% or so of people are ultimately going to get type 2 diabetes. That's a lot of people. Obesity is a major risk factor, and with the obesity epidemic in the U.S., this is skyrocketing, and everywhere else in the developed world and in the developing world. And it's heavily genetic. At least 50, 60% of the risk of this, and some studies maybe as high as 80% of the risk, is determined by your genes. But there are very, very rare, there are a few single gene disorders that cause this, but they're extremely rare. And here, all at once, a whole bunch of studies hit and found stuff. And there was enormous excitement because this had been completely intractable until now. And I'll tell you in a minute just how predictive those risk factors are, but they are real risk factors. So the statistics here is complicated, and I'm not going to talk much about because I don't understand most of it, but I'm going to talk a little bit about the basic problem here. And this is this multiple observation problem that I talked a little bit about already. Imagine if you roll the dice, your chances of getting 2 sixes, 6 on each die, or 36, is 1 in 36, or 2.8%. However, if you roll the dice twice, the chance that one of those rolls will be 2 sixes is much higher, 5.5%. If you roll the dice 100 times, the chances that you'll get 2 sixes one of those 100 times is 94%. So if I simply say, look, I rolled the dice and I got 2 sixes, isn't that remarkable? Well, it's not that remarkable if you've rolled the dice 100 times. Well, think about when you do a chip, a SNP chip that has 1 million SNPs on it. You've basically rolled the dice 1 million times. So what is significant. How different does the patient group have to be from the controls to be significant? And in reality, what ends up happening is we base this on p-values. The statisticians basically calculate a p-value. But instead of 0.05, a 1 in 20 by chance, that's obviously not good enough here. Generally, the cutoff for being significant in these studies is p-values of 10 to the minus 6, 1 in a million, to 10 to the minus 8, 1 in 100 million. And that's what these are, p-values. So somewhere around here is the magic line. You can see if you look at the statistical significance of the millions of SNPs along here, plenty of them are significant even at 1 in 100,000, purely by chance. But some things clearly stick out. So you'll see this is the data for an accumulation of the data for type 2 diabetes from a few years ago. And you can see this is the biggest hit, TCF7L2, a transcription factor, and it's got a p-value of 10 to the minus 50. That's a big number for you, those of you who like statistics. 1 in 10 to the 50th. I don't know what that is. There's probably not a name for it. It's beyond petabytes. I don't know what that number is. And actually, tell you the truth, I'm actually not that sure that the sun is going to rise tomorrow. This is quite a high statistical number. I mean, I'm pretty sure the sun's going to rise tomorrow, but I'm not sure that number's at 10 to the minus 50th. So this is real. In fact, all of these are real. These truly are genes that affect your risk of getting diabetes, of type 2 diabetes. And as you can see, there are quite a lot of them. All the ones that are highlighted in color here are truly significant. These are variants within the human genome that contribute to your risk of diabetes. So this is enormously exciting. Here we have been trying for years and never identified anything. Certainly not this gene. And actually, almost none of these were even candidates before. And now we've got 20 or 30 genes where variation in those genes contributes to diabetes risk. Well, is that useful? The short answer to that is no, not clinically useful. It's fascinating scientifically. Some of these genes might be interesting. Maybe they'll give us clues. Maybe we'll develop new treatments based on them. A few of them actually turn out to be, not many, a couple are actually targets for drugs that we currently use to treat diabetes, but not many of them. And the hope is maybe they'll identify other targets. Who knows? But is it useful? Well, it turns out, you know what's really useful? If you look at body mass index, or your weight, that turns out to be extremely predictive. If you're overweight, if you're in the upper quartile here of body mass index, you have a very high risk of developing diabetes, and it's much lower. It's about 4-fold higher. You got about a 5% lifetime risk of getting diabetes if you're in the lower quartile of body mass index, and almost a 25% chance if you're in the upper quartile. And if you have a positive family history, that increases the risk a little bit too. Now keep in mind, these diabetes risk variants, they affect your risk by 1.1-fold, 1.2-fold. This is 4-fold. So this is a paper from the New England Journal that sort of reviewed this, actually did a clinical study to try to determine, is this kind of genotyping useful? And their basic conclusion was no. And sort of summarized here. These two tests, highly sophisticated tests that we've only been able to do for several hundred years actually, are far more powerful than this somewhat expensive genetic test. If you step on the scale, that will tell you more about your risk of developing diabetes than all of this testing. And if you add in family history, it's even better. By the way, we very rarely in clinical practice take much of a family history. That's a disappearing art. But if you take a good family history, it's actually useful. Okay, well, have genome-wide association studies been done for anything else? Yeah, you sure have, for hundreds of diseases now. And I know Dr. Gallardo, I'm going to show you one of the same compilations from the NIH that Dr. Gallardo showed you. But here's a list of some of the big important diseases, just a few to highlight, for which these kinds of genome-wide association studies have now been done. A biggie, of course, is heart disease, heart attack, stroke, atrial fibrillation, inflammatory bowel disease, asthma, a whole variety of neuropsychiatric diseases, depression, bipolar disorder, ALS, multiple sclerosis, schizophrenia, Alzheimer's, a whole bunch of rheumatologic diseases, many different cancers, and even a whole bunch of common traits. One of the most heritable traits among humans, by the way, is height. Height is about 95% heritable. You look at your family history. It's not entirely predictive, but you get a rough idea of how tall you're going to be. If you have an identical twin, look at them and you get a very good prediction of how tall you'll be. And large GWASs have been done for height in, I think it's over 100,000 individuals now. And some clear-cut genes have been found that are somewhat, that do affect what your height's going to be. And if you take them all together, you can explain about 10% of that heritability. The other 80%, we still don't know what it is. And it's still quite a mystery. Why aren't we finding it? We've looked at 100,000 people. Some people refer to this as the missing heritability or the dark matter. It's something genetic. We don't know what it is. Most of us think it's probably much rarer variants. All you can find by a GWAS is things that are very common. It has to be pretty common if you're going to be able to compare your patients and your controls. If just 3 of your patients have it and none of your controls, you're not going to pick that up. So if there are thousands of things that only a few people have, you're not going to find it easily this way. And we think that's one of the possible explanations. Another is it may be complicated interactions between genes. Well, what if this thing doesn't do anything by itself? But if you have this SNP plus this SNP plus this SNP, that does. Well, if you think looking at 1 million SNPs is a multiple observations problem, what about 1 million times 1 million? So we just don't know how to deal with that mathematically yet. So there's a lot still to figure out. And we may get much better at it, but right now, we're not very good. This, I think Dr. Glarder showed you, this is a compilation of the diseases for which GWASs have been done, and we now, have loci like this. This is, there's many more than this. This is from 2 years ago, but here's where those various variants are that are associated with these diseases. Well, they're all over the place. You'll notice there aren't many on the X chromosome. Now, isn't that odd? There aren't any on the Y chromosome. That's actually not that odd. I don't know if it's sort of embarrassing as a man to point this out, but there's actually very little of redeeming social value on the Y chromosome. There are very few genes at all on the Y chromosome. And you can see it's pretty little and meaningless. All the action is here. A pretty significant part of your genome resides on the X chromosome. So why haven't we found anything here? This is actually sort of a little dirty secret. Because of the way GWASs are done, we generally don't look at the X chromosome. So if there are, that's another potential source of missing heritability. If there's an important variant on the X chromosome, we've probably missed it at this point. What's that? No, the X chromosome. Because of the statistical analysis, it's a little more complicated. Men only have 1 X, women have 2, and it's generally left out. When you do a GWAS, you look at all these chromosomes, and you just don't look at many SNPs on those. So if there were something there, we're missing it. X or Y. Y, it wouldn't matter. There's no point in looking at the Y. We know why. No, I shouldn't say. Y is responsible for some things. As all of you know, I probably, by now you figure that out. The Y chromosome does, it does determine gender. It plays a role in determining gender. And there are a few other things. There's a great map of the Y chromosome that one of my colleagues, Jane Gitcher, published a number of years back. The gene for channel flipping, for example, is thought to be on the Y chromosome. Inability to ask for directions. There are a number of things like that that have been mapped facetiously to the Y chromosome. But there really aren't many genes that are going to do it. But the X is a source of enormous contribution here, and it's really not been adequately looked at. Okay. So what have we learned from all these GWASs? I don't want to trash this entirely. I think some of this is useful, and I gave you a few examples where we have found useful things. But one of the key things, and I already mentioned this, that we've learned is that almost all of these candidate gene association studies that were published previously, about 40,000 of them, the vast majority of them are wrong. The optimists say only 90% of them are wrong. I think it's closer to 98%. You could debate. But for example, if you take diabetes and you look at the several thousand single gene association candidates that were done prior to the GWAS era, and many were reported to be strongly associated. If those were right, they should have shown up on the GWASs. Almost none of them did. So that's how we know they're wrong. Most of the variants have a very small effect, 1.1. They'll increase your risk of diabetes from the population risk of 15% all the way up to 15.5%. Now, is that clinically useful? Not really. And overall, if you put them together, we just can't account for all that much risk. It's just not that, again, you're better off stepping on the scale and taking a family history. Are they going to identify all kinds of new biologic pathways, new drug targets? There's lots of optimism. We don't really know. There's not lots of success yet. There are some exceptions. There have been a few studies that have identified big effect genes. There's the example of age-related macular degeneration. There is a common variant found by GWAS that accounts for a big chunk of your genetic risk of getting AMD. Thrombosis, factor V Leiden. Factor V Leiden actually was not discovered by GWAS, but it would have been had we done a GWAS for it. That's why I listed it there. It's a single common variant that has a pretty big effect and accounts for maybe 25% of the risk of having a thrombosis. This is probably one of the best. It's a little bit obscure, but it's relevant to all our talks about the hemoglobinopathies a few days ago. This is a GWAS was done for fetal hemoglobin levels. You know, we all have a little fetal hemoglobin, 1 or 2% or less in our blood cells. So they simply collected a whole bunch of people, measured their fetal hemoglobin, and did a GWAS to see if they could find some gene that affected the variation in fetal globin levels in the normal population. They found a variant in this gene, BCL11A, that had a really big effect on people's fetal globin levels. Well, guess what? It turns out to be protective if you have thalassemia or sickle cell anemia. What's more, it turns out this gene, it's a transcription factor. If you knock it out in mice, they totally lose the ability to do the fetal globin switch. So we found, the people who did this, actually my former mentor Stu Orkin at Children's Hospital in Boston, found that this gene is the basis of the fetal hemoglobin switch, or a really big chunk, which had been a holy grail in the scientific field for many years. And this all came from studying, from a GWAS study. So there are going to be big hits like this. There are going to be some conditions for which a common variant is going to have a big effect. The key limiting thing of a GWAS is it's only going to work if a very common variant is having a significant effect. And that just doesn't happen all that often. And we can't tell in advance, but for some conditions it has. There are a few infectious diseases, responses to drugs, where GWASs have had identified big effect genes, but they're relatively limited. So this brings us sort of to the next. So when are we going to use this stuff? We've got lots of tests we can do here. We've got all these variants we've identified. We have some gene mutations we've identified. When should we actually use this stuff? You know, you got a genome with 3 billion base pairs. You could look at any of those. You know, you have more than 3 billion little tests you could do here. When are you going to order them? When you're sitting in the clinic? Well, I would urge you to sort of think this way, and this is actually how geneticists think. And I've sort of divided the kinds of tests you could do into 3 groups. What I'm calling class 1 here are tests for which the result of that test has a clear-cut impact on how you're going to treat the patient. If we have result A, we're going to give the patient drug X. If we have result B, we're going to give them drug Y. To me, that test is clearly indicated if they're going to impact the way we manage the patient. And I should point out, this doesn't only apply to genetic tests. This kind of reasoning should apply in your medical practice to everything you do. Do you order an X-ray? Do you order a blood count? You should be thinking, is this going to change the way I manage the patient? Because if it doesn't, it might not be worth spending the money. And this is, of course, as you know, is an enormous problem in the practice of medicine. We do an enormous number of unnecessary medical tests that don't fit into class 1. In fact, the vast majority of tests we do probably fit into class 3. And it's an enormous, if we could eliminate this kind of testing, we would cut our current medical expenditures probably by a factor of 2 or more. It's a big issue and a big problem to think about. Yeah? Yep, absolutely. And that's a very valid. So the question is, class 3, if it doesn't have an impact on medical management, the patient might still want to know. And that's absolutely valid. But then you have to think about it a little bit differently. So Huntington's disease or Alzheimer's predisposition are the classic examples of just what you're talking about. When we see patients in the genetics clinic who are at risk for Huntington's disease, have a father with Huntington's disease, they've got a 50-50 risk of having Huntington's disease. If they've got the gene, they're going to probably develop a severe dementia and die in their 30s or 40s. Well, that might affect your plans to have children. You know, what kind of job do you want to get? Do you want to keep going to school till you're 50? You know, do you want to go to medical school and finish your residency and get a job when you're 45? You know, that's going to have an impact on you, right? Absolutely. But there's no it's purely a personal choice. On the other hand, You might not want to know that information. Some people would say, geez, if I know that, it's going to totally screw up my life. I might be paralyzed and never do anything, and then by the time I'm 30, there's a cure for Huntington's disease, and I should have ignored it. Maybe I don't want to know this. So what we do here is the problem is once you do the test and give the information to the patient, there's no taking it back. So in genetics, we spend a lot of time counseling patients. Do you really want to know this information? And I don't know if this has been discussed. In the case of Huntington's disease, when we counsel patients about it, say, okay, do you want to be tested or not? What percentage do you think decide to be tested? 25%. I used to do the patient presentation to you guys on Huntington's, and I brought in two sisters who were both at 50-50 risk for Huntington's disease. One decided she wanted to be tested. The other side she didn't. The one who got tested was positive and had Huntington's disease, and she eventually died of it. And but they used to come and talk to you guys and explain. They were both satisfied with their decision to be tested or not to be tested, and they talk about their reasons for it. It's a very personal thing, but it's an important thing to think about. On the other hand, as a physician, if there's clearly going to change my medical management, we don't need to spend a lot of time discussing, do you want to know this or not? You should know this. It's straightforward. Here, it's a very different thing. And of course, there's this whole big area, like everything else, where is it of help, is it not? We're not so sure. But this is a critical issue, and much of the future of medicine, comparative effectiveness research is going to be figuring out which tests are in this class that we should do and which should we leave to the patient. And we may come back to this at the very end because I think we do everybody's genome. We're going to know all this information. It's going to be up to the patient to decide what they'd like to know and what not. So I was just going to give you another example here, the factor V Leiden, which I've discussed before, a very common, the most common genetic risk factor for thrombosis. About 5% of the population has it. Venous thrombosis is highly heritable, where you're going to get a blood clot, which can go to your lungs, a pulmonary embolus, a very common cause of death in the U.S. And it's highly heritable. About 60% of the risk is genetic, and about half of that, close to half of that, is this factor V Leiden. There are a few other things as well. There are a bunch of other risk factors, but this factor V Leiden is present in 5% of the population of European origin. These tests are commonly ordered in clinical practice. In fact, I would one report that factor V Leiden is the most commonly ordered genetic test in the United States today. And many of these others are ordered. They're much less common. They're also risk factors. In addition, I think it's worth pointing out that there are a couple of other risk factors that are bogus. These were the single gene associations that turned out just plain to be wrong. These are clearly now not risk factors for thrombosis. They are still tested for many places incorrectly. There's a huge amount of misinformation. In fact, this was actually offered for quite a while on the menu of tests at the University of Michigan. I used to see patients referred to me in clinic who were very upset that they had this polymorphism, and they were told they might have an increased risk of thrombosis. There are a few studies that say it increases your risk. There are a couple of studies that say it decreases your risk. And on balance, in the really good studies, it has no effect at all. And I made quite a protest, and they finally agreed to take it off the menu of tests at the University of Michigan, so none of you can order it. But they left it on the menu for reference outside labs because doctors order it, and they don't want to lose the business to another lab. That's how medicine works. Don't order these tests. But should you even order factor V Leiden? Well, here's the key thing to me. Does it change your treatment? So here's what we do if a patient has factor V Leiden or if they don't. When they have a blood clot, we give them a blood thinner. And then we keep treating them for 3 to 12 months. If they have multiple blood clots, if they don't have factor V Leiden, we keep them on a blood thinner long term. If they do have factor V Leiden, we keep them on a blood thinner long term. Bottom line is it doesn't change our management. There's a couple of conditions where we really aren't quite sure what to do, but there's clearly no clear-cut evidence that it should change our management. As a result, I personally never ordered this test. Actually, I ordered it once, and I regretted it, and if any of you want to hear the story, I'll tell you afterwards. But I never ordered it. And again, this is the most commonly ordered test in the U.S. And this is my particular area of expertise, inherited bleeding and clotting. I never ordered it, and most of my academic colleagues never order it. But it's widely ordered in practice. And again, it fits into this what do we do about it category. And that's why I don't order it. Okay. So again, important concept to think about when you want to order these tests. So let me switch a little bit. It's not really switch, continuing in a way, talking about personalized medicine. I have to sort of start out by telling you, I hate this word. And you'll hear it used all the time, and you'll read it in the newspapers. Personalized medicine, as if this is some exciting new thing that we've never done before. And I have to tell you, I am offended by that as a physician. I've been practicing medicine for, 30 years or something, and I think we've always been practicing personalized medicine. When I go and see a patient, I take their history. I don't take the general public's history. I examine that patient, I order tests on them, and then I come up with a very personalized diagnosis and a very personalized treatment. I write out a specific prescription for that patient, and I think that what we do in medicine is very personalized and always has been. The other reason I find this offensive is it sort of implies that impersonalized medicine is a bad thing. And that's actually also not true. Non-personalized medicine is public health. Clean water, vaccinations, this kind of stuff. That's impersonal medicine. You know what? This has had a much greater impact on human lifespan, survival, quality of life than any of the personalized medicine we've been doing for 50 or 60 years. So I tend not to use that term, but you will see it widely used. And I hope at least a few of you at least snicker a little bit when you read personalized medicine. So what do we really mean when we use the term personalized medicine? I think what we mean is harnessing the power of genetic information to improve treatment, tailor treatment, identify disease susceptibility before illness comes, facilitate preventive treatment, give write better prescriptions that are less likely to cause toxicity. Several years ago, this was sort of the holy grail, was to develop tailored medicines, that we do a genetic test in a patient, identify their disease, and give them just the right specific treatment made just for their disease. And the sort of poster child for this was a drug called Gleevec, which you'll all learn more about at some point. This is a tailored drug that specifically hits a disease called CML, chronic myelogenous leukemia, that has a very specific acquired somatic mutation. There's a chromosomal translocation between chromosome 9 and 22, makes a new unique gene, a BCR-ABL fusion protein that's only present in CML, not in normal cells. And this drug specifically targets this, and it's almost curative, and it has completely revolutionized the treatment of this disease. Median survival used to be 3 to 4 years. Now we don't know what it is because these people seem to be living relatively normal lifespans. A miracle. It was remarkable. Covers of Time magazine, etc. Everybody's excited. This is the future. Well, we've not really been able to duplicate this yet. This is sort of a one-of-a-kind phenomenon at this point, though we're all hopeful that we'll do greater things like this in the future. There are some examples of biomarkers where we have a genetic test that helps us predict a bit who's going to respond to particular types of treatment or who won't, various toxicities. But so far, very few of these are used. There are a few that are slowly sneaking into clinical practice, but not many. One of the best that got a big flurry of interest initially this is a GWAS for responses to the drug Coumadin. And you can see some really dramatic single peaks. It's not lots of little things like diabetes, and these turned out to be really big and very satisfying. This turns out to be the protein that Coumadin interacts with to work as a blood thinner. It's the target of Coumadin, and there's genetic variation in this that determines people's response to Coumadin. And this was all very exciting. It was so exciting that the FDA actually suggested on the label for Coumadin that you consider genetic testing for these factors in choosing your dose. In my view, that was quite premature. Very few people do this testing. The bottom line is, yes, it's predictive. You can predict about 30% of the variability in response to Coumadin with these things, but it's just not that useful clinically because the other 70% is pretty important. And in reality, what you do is you start a patient on the drug, and if they don't have a good response, you increase the dose, and if they have too good a response, you lower the dose. And by the time the test result comes back a few weeks later, you already know what dose they should be on. And in practice, it's just not used. Its clinical value is certainly unproven, and it's just not used clinically. And again, there are others that have potential and theoretical use. One of the best is a variation, a common variation in a metabolic enzyme that affects a chemotherapy drug that's given to treat a whole variety of autoimmune diseases and some cancers. And there are some people who are very, very sensitive to this drug and have very severe reactions. and it's been proposed that we should be screening patients before we put them on this drug. It's just not done much in practice, and here's a sort of depressing review from a clinical journal reviewing all the published data, and there's just not convincing data that this test is worth doing. And you do sort of a risk-benefit analysis. And the plea I would make is we need this kind of data before we start doing these tests in the clinic. We need evidence that they're really useful, and we just don't have that much yet. Well, the last thing I want to talk about in the last couple of minutes is direct-to-consumer genetic testing, because you guys will hear about this and see this, and your patients are going to come in with the genetic analysis they had done outside and say, here, doc, here's my genome. What should I do? And after this course, of course, all of you know exactly what to do. So I already sort of convinced you, I hope, that this is useful for Mendelian genetic disorders. Not all that useful for this other stuff. I'll just mention as an aside, well, patients can go get this. You can order it on the internet. It's about $300 or $400 to send a cheek swab off to 23andMe, and they'll give you back a report of your million SNPs, and they'll tell you that your diabetes risk is increased and your risk of prostate cancer is decreased, and they'll give you this long, detailed report. And is this useful? What do you do with it? This was a study done sending the exact same sample off to a bunch of different labs. They all got the same SNP results, but they all interpreted them differently. One place would tell you your risk is decreased. The same person's risk in another lab would be estimated as increased. Bottom line is it's just not that useful or predictive. And that was sort of the conclusion of a General Accounting Office study. But nonetheless, this is available and widely used, and the major evidence to support it are all kinds of testimonials. If you go to the website, this changed my life, et cetera, et cetera. But in reality, if you look for hard evidence that this really has a big increase, a big effect on what patients do in their lives, I can tell you that the current evidence, the hard scientific evidence, is summarized on the next slide. There's none. So, in terms of how useful it is to the public, personally, I would put it in the same realm as astrology and tarot cards. People like that. And it's probably not any more harmful than those are. And, it's part of this large universe of things out there that are available to patients of dubious medical value. And I would put SNP genotyping in the same category, at least for now. Nonetheless, your patients are going to bring in these reports. Yeah?
Is it actually the same, like, genotyping or, DNA sequencing if you came to, like, say, the period aspect of that?
Good question. What I'm talking about here is just the SNP. We're just going to take those million common variants. We're not looking for disease mutations. We're using a million common variants, and then we'll just tell you, based on that, your diabetes risk is 15.4%, not 14.9% like everybody else, and that kind of thing. It's those kinds of SNPs predominantly. They do put a few common disease mutations on them, like this factor V Leiden. So you will end up finding out your factor V Leiden status, a few other things. But they're not doing sequencing. That's coming. And that will be something patients will be able to order. And by the time you guys are practicing, they're going to come in with their whole genome. That'll be a different matter. Because if they come to you and say, well, I just found out I have Huntington's disease, what should I do? But you're going to probably have to talk to them about that. You can't dismiss that as easily as you can dismiss this stuff. This, I'm just talking about the minor common variants that everybody has and the different little mix you happen to get. There are other recreational things you can do with this testing, and it's extremely good at this, I should say. This kind of SNP testing, if you get half these variants from your mom and half from your dad, and if you look at a million of them, we can tell you with a p-value of 10 to the minus a million who's your mom and who's your dad. So if you'd like to know that, moms, by the way, are rarely a question. But dads often are. At least a few % of people, their father is not who they think he is. And if you'd like to find that out, you get a cheek swab, and you got to get dad to volunteer to give a cheek swab, and you can find out. You can also find long-lost relatives. They are very good at telling you your ancestry. You're 15% African, you're 13% Native American, you're this. That from this analysis is actually very, very accurate, and a lot of people are finding this very interesting and entertaining. That's valid. That's much better than tarot cards. This is actually accurate and correct, and usually better than your own, what you think your ancestry is. And you can get that, and people are really into ancestry. What's going to eventually happen, and they have databases of millions of people, when you sign up for one of these, if you do send your cheek swab off to 23andMe, one of the things they'll ask you is they say, well, would you like to know if you have any other relatives who are in here? And if you give your consent, and they have, so I have a friend who has an adopted child from Guatemala, and she did him, really just as an experimental thing, and got back a thing and they found some relatives who were, second cousins or whatever, and you can figure all that out with this kind of analysis. So that sort of stuff can be done this way. Major thing I want to close with is that next-gen sequencing, which I've mentioned several times, this ability to sequence incredible amounts of sequence incredibly fast, is revolutionizing everything. This is a slide that shows you the cost of DNA sequencing. And this is Moore's law. Some of you may know this is this computer thing that the cost of storing information and computing power has sort of dropped logarithmically. Well, the cost of sequencing is dropping much quicker. It's really astounding how cheap it is to sequence now. In fact, this gap is a problem. The cost of storing the sequencing data is getting to be a real rate-limiting thing, more than doing the sequencing itself. And I like this slide, which I got from Eric Green at the NIH. This is Francis Collins, who used to be at Michigan, is the head of the NIH. This is what you could get for $10 million when Francis was the head of the Genome Institute in 2000. It cost about $20 million to do a genome. And this is Eric Green, who's the current director of the Genome Institute, and Eric likes to show this is how many genomes you could do in 2010. It's a graphic illustration of the drop of sequencing costs. So the final thing is just sort of speculating about where all this is going to lead us, and things are going to be, the only thing I tell you with certainty is by the time you guys are practicing, things are going to be dramatically different than they are now, and certainly than they were when I started. I suspect that we'll be doing newborn screening by whole genome sequencing. It'll be cheaper to do that than screen for the 30 or 40 diseases we do now. We'll probably only look at those actionable class 1 things that you want to know about early, but all the others will be there. And if a patient wants to know, they'll have the right to know that, and it's going to be a hard thing to figure out. We're going to get better therapies. We're going to get better at predicting risks. And we're going to have large data sets of everybody's genome and everybody's health information all available for research purposes, and we're going to learn enormous things. It's going to be a very exciting time. So I will stop there. Wish all of you luck, and you got to stay on top of this stuff because it's going to be important.